In today's video, we're going to be talking about the anatomy of the vagus nerve, which can help you potentially explain why this nerve is so incredibly powerful, why it might be implicated in more than 100 human diseases, and also more importantly, why different individuals perhaps could respond differently to the vagus nerve neuromodulation device that I've been talking about in my videos. Speaking of which, this video is sponsored by Parasim. That's the company that makes Neurosim, which is one of the earliest to the market vagus neuromodulation devices that we know of. This is great. And I will tell you by discussing what the vagus nerve looks like, why some people might respond very well and others not so well. All right. If you're wondering why not everyone can potentially benefit from such technology, this can help you understand why. My name is Dr. Mikhail Rashek of Microscopic. Let's get started. By the way, also check out the link in the description so that you can, if you're interested in this device, you can get it at a discount. All right, so let's get going. So this paper I want to be discussing today, they literally looked at vagus nerve. They cut it, like imagine you cut across it and then you look basically from the top view what it looks like inside. That's what we're going to be talking about this. But what the authors mentioned is that, look, vagus nerve is not enormously deeply studied, and the reason why is because you can get a biopsy from a living individual, so most of the studies come from cadavers. And here's an issue with that as well on its own right, and the reason for that is because what is happening there is that oftentimes, of course, tissue can already degrade, and on top of that, oftentimes this comes from elderly individuals who, as a consequence, might have already had certain deterioration of nerves happening. This is due to aging. You can have neurodegenerative diseases, but also something like even and diabetes, for example, they mention is an example of that, where the older you are, the more likely you have the risk of experiencing diabetes. Diabetes can have an indication of neurodegeneration as well in the peripheral nervous system. They mention also cancers. Cancers in their own right can affect your nervous system and lead to its degradation, but cancer treatments can achieve that as well. So you don't have necessarily perfect and intact samples to work with to get really deep understanding of how these nerves look like and how they function. And we need to understand this, especially now in the new era of bioelectronic medicine, where we have the capacity to potentially influence our health by directly activating nerves, such as the vagus nerve, via the device that I've been talking about. So what they proposed and what they did is they use organ donors, and the reason for that is because, number one, typically tissues have to be handled differently. So in such a case, the tissues are still oxygenated and therefore they are maintained intact. And in this case, they also used young people. These were people who were pronounced brain dead and harvested for organs to basically save human lives. And this allowed these researchers to look at the vagus nerve. And they looked at vagus nerve in a group of young people, mid-40s is when, unfortunately, when they died. And they looked at both the cervical component of the vagus nerve, which is near your back, near your neck, as well as abdominal vagus nerve, which is, of course, one that is now in your abdomen, as the name suggests, so deep down. Speaking of which, because remember, the vagus nerve goes from a brainstem and travels through your neck, and it then explores your entire body, all the way through your heart, through your lungs, liver, all the way to your abdomen. And in studying for this video, I found this very cool image that one researcher showed, where they showed the path of the vagus nerve, and you can appreciate how big it is and how massive the vagus nerve actually can be. So they looked at the one near the top, that is close to the brainstem, and one that is that is in your abdomen. So we're going to basically discuss how such vagus nerve components, how can they differ from one another, that can help you understand how it works. So first, we're going to basically look at light microscopy, basically regular microscope image. So you take the nerve from this area, somewhere around your neck, and remember, we're cutting across it. So we're going to look at from the top of what it looks like inside. And they use specific type of stain that allow them to see fatty substances. So they allow them to see myelin layer on the nerves. We're going to talk about this, explain that in detail as well, what that is. Myelin is this protective layer on top of nerves, which it can with different stains, it might not even be visible under microscope. So they use specific stain in order to achieve that. And here I gotta warn you though, if you have a phobia of looking at many different dots or many different bumps, this might be triggering, because people who have this phobia, I think it's called like something like trypophobia or trypophobia, something like that. People who cannot handle sight of many holes in one area, it makes them feel nauseous or sick, or just think simply they feel anxiety, then you might not want to watch this video. The reason why is because we're actually going to be seeing neurons cut across nerve cells, axons specifically. Axons are the long extensions of nerve cells that travel through your body. We're going to be seeing that, and you'll see how you can package many of neurons together in like almost like a cable. And that's what neurons are. They basically, imagine cables running through your body that are sending current. And when you see many of these, you see these many little circles. And for some people, that could really be upsetting to see that. But if you're not that type of person, you don't have that phobia, then enjoy, because you're going to see very cool anatomical pictures of the vagus nerve, one of the most powerful nerves in our body, and one of the most powerful nerves in our body in the context of our health, and how that nerve itself can help in health benefits. So they stain this nerve with a special stain, and when you cut across it in the image, what you will be able to see, so you see it's the nerve itself is, you can see these darker structures inside. First, actually, maybe talk about these black circles, or dark blue circles, I should say. These are fat droplets. They're lipid droplets. Authors don't explain as to why they are in the vagus nerve and what their role might be. I couldn't find that information, so I won't discuss this. But then on top of that, you see these very much bigger structures that have nerve cells. They have these little dots inside. Those are the nerves, the axons. And these structures are called fascicles. So basically, think of fascicle like a cable that has many wires inside. Fascicle is an interesting word. It comes from the word fasces, which is bundle of sticks put together. So you can see how bundle of nerves derive its name from that word, and hence how you got the name of the word fascicle. But of course, fasces also meant that bundle of sticks is a symbol of authority. So that's why you might also hear it in other contexts as well. But that's where the root of the word comes from. So when I talk about fascicles, I'm talking about bundles of nerves. And you can see these bundles of nerves there. And there's numerous of these in the vagus nerve. You can see also the scale bar there is 500 micrometers. So the entire nerve is few millimeters in width. You can see it's kind of flattish. And between all these fascicles, you have a lighter color. That's basically a connective tissue that connects the entire vagus nerve together. It's called Perineum. And it's mostly made of collagen, but it will also have blood vessels. Obviously, you need to bring blood to the nervous system for them to function properly, and of course, just to work normally, you need a blood supply. And then the authors look specifically deeper into those fascicles themselves. Okay, and And this is where it gets really, really interesting. So you can see that these fascicles are surrounded by this protective layer, protective sheath. That's called perineum. So then that's made out of specific cells, and it obviously it protects the nerves, the bundle of nerves themselves. So it protects the fascicle, okay? And they show, you can see them, the authors point them out with colored arrows in these images. And they zoom in on those fascicles. This is where it gets really interesting. Once you zoom in into these fascicles, you see a bunch of little circles. All these circles that you see, those are all individual nerves. And by the end of this video, you'll find out that vagus nerve basically distributes thousands upon thousands of nerves into your entire body. So you just wait for the details, okay? And you can see all these circles. You can see these circles, they have this dark, dark rim. That dark rim you see is the myelin layer, the protective fatty substance that many neurons are coated with. What the myelin achieves, not only does it protect the neuron, obviously, it also achieves that the electrical impulse travels faster through the nerve. It almost also ensures that the power of the electrical impulse doesn't dissipate through the nerve, so that myelin acts, almost think of it like a wire rubber protection as well. So that's what myelin achieves. It allows the nerves to basically communicate faster. And you see many of these in these fascicles, right? So, and we're gonna get to the bottom of this as to what their different roles in the context of the vagus nerve is. Now, also one more thing I'll tell you before we move on is that fascicles also carry both afferent and efferent nerves. Afferent meaning nerves that travel from your brain to the body, and efferent or efferent nerves are the ones that travel from the body to your brain. So a single fascicle can have both highways of communication in it, while they're obviously going to be responsible for different roles, and the communication goes in different directions. And that's basically how your body is in a constant dialogue with your brain. Okay, then let's now look at another of their light microscopy images with this stain, and this basically now you're looking at another fascicle in detail. And you can see that perineum, now you see it in greater detail. And if you look at it closely, perineum, that protective layer around the nerves themselves, it's like packed with many layers, or many basically sheets of cells are building it. So that's called lamina. So, and you can see that it's many layers of cells that are building this protective layer. In the image, the authors pointed out with the arrowheads. So now that you got a good look, a detailed look at fascicles, let's move on to the next type of images that the authors made, which is really, really incredible. Now we're going to move to electron microscopy. So light microscopy was the typical microscope you see in every movie. Wherever scientists are presented in movies, there's always obligatory microscope on a desk, right? That's light microscope. Electron microscopy, you never see it in the movies because it's more like a box because it's a very powerful, complex instrument that allows you to see now on a microscopic level, almost on an atomic level. This is as close as we can get to the atomic level at the moment in terms of taking pictures. We literally bounce off electrons off of the three-dimensional surface, and that is what we're seeing. And again, we're looking at a zoom in at inside of the fascicle with the electron microscopy. So these are the black and white images, and what you can see now, you can see both the myelinated neurons and unmyelinated neurons. So the myelinated neurons, again, are the ones that you see this very dark rim around the neuron. So you can see the myelin protecting the neuron, and then there's a number of different neurons that are not protected with that sheet at all. So these are the unmyelinated neurons. I know I'm gonna mess it up saying these words sooner or later. And they zoom in on that, so you can see see greater detail. What the authors got really excited about is that how well the tissue was preserved, so you can see such fine detailed structures. When you look inside the neurons with the electron microscope, you can literally see the cytoskeleton inside these axons. Remember, these are the part of axons are the long extensions of neurons that allow you to transfer information long distances from one area to boom all the way down. So you see these axons cut across, and you see literally cytoskeleton. Cytoskeleton is a structural component that basically is like, think of skeleton inside the body. This is a skeleton inside the cell. It's made out of neurofilaments. Neurofilaments are more like permanent structure, so that truly more resembles a skeleton. And it also is made out of microtubules. And microtubules also act like a skeleton, but they're more dynamic. They can change paths. They can be built up rapidly and rebuilt. And those are actually more like highways inside the cell. So these microtubules, you can attach robots on top of them, and they act like train tracks. And then robots literally travel along those train tracks. Molecular robots, obviously, inside the cell, they travel along those microtubule train tracks and move things around the cell, including, for example, vesicles that have neurotransmitters inside of them. So neurotransmitters are the chemicals that a nerve cell releases at the end in order to send information across space from one nerve cell to another. This is how our brain works. So then these vesicles containing neurotransmitters, they literally can be moved along such train tracks, and you can literally see these tiny little puncti in these images. That's what you're looking at right there, as well as certain organelles. You can see the darker little circles inside these neurons are mitochondria. So mitochondria are the factories of, they're like coal factories, think of it, in on our planet, or nuclear power plants, or wind farms, whatever you want to use as an analogy. They're the ones that produce an energy for our body. So they're very key, obviously. And you can see fragments of them, because remember, this just cut across, and the authors point them out with black arrows in this image. And yeah, so they were like super excited, right? And then I think what else we're looking at inside there. Okay, now let's move on to, I think, Schwan cells. Then there you can see these unmyelinated neurons as well in there, right? But around them you see these structures that they called Schwan cells. Now, Schwan cells are really interesting type of cells because they're very important in making sure that your nervous system is healthy. These Schwann cells produce that myelin layer for neurons. It can also degrade it if it gets destroyed and regenerated. And Schwann cells also can help neurons regenerate as well. Myelin, by the way, if I'm not mistaken, that's the component that can be destroyed in certain conditions that lead to certain neurological conditions, such as multiple sclerosis. So that's one example. Another example is the Schwann cells. They can also be eventually stop functioning as good, and that's a hallmark of aging. That's one of the reasons why with age you stop functioning as lucidly with your brain as when you're young, because eventually things degrade. So the Schwann cells can also degrade over time with aging, and that can exhibit the decline of mental capacity, for example. So but the Schwann cells, what they do in these images, what you can see is they start surrounding those unmyelinated cells, the ones that don't have myelin, and Schwann cells can bundle those up together. Now, when the Schwann cells and these unmyelinated nerves are bundled together, that has its own name. Forgot what it's called. It's a weird name, like. I'm not gonna try to remember because I'm not gonna mess it up. But basically, it's this layer that the Schwan cells release. Those are made of proteins. It's like a dense protein matrix, and that acts as a protection for the form of protection for the unmyelinated cells. And that's why oftentimes you see Schwann cell near a bunch of unmyelinated cells. So, and I think authors pointed out with like the color arrows. So one more thing that the authors did that is very cool in these electron microgram images is they zoomed in super duper close on the myelinated layers. And again, you can see how it's like that lamina where there's layers stacked of like basically biological material, like sheets. You can see that one after the other. So you can see how the myelin layer actually works in protecting the nerve cell. And it's this layer that can be destroyed in certain conditions. So, and so that was all about the cervical nerve. So you can see once you start zooming in with the electron microscopy, you see not just the myelinated cells that were very easy to spot with with the light microscopy. Now you see these unmyelinated cells. And now, so let's now look at the abdominal part of the vagus nerve, and we're gonna go back to the light microscopy again, okay? So now we're just regular microscope, stained with that blue stain, and now you see the big thing you'll see, then there's fewer fascicles inside the abdominal portion of the vagus nerve. That makes sense. As the nerve was traveling down your body, it kept releasing branches to innervate different organs. So different fascicles basically already started exit the vagus nerve pathway to start connecting to specific organs such as your lungs or your liver, and there will be fewer fascicles as you go into the abdomen. But abdomen is still one that has to be crazy innervated, as you saw in the picture earlier. And otherwise they look the same. Like there's still these lipid droplets, there's the connective tissue, just fewer fascicles. And they zoom in on those fascicles again. Once again, they point out the perineum with the colored arrows in those images. Perineum, remember, is the protective sheet of cells around fascicles. So think of it like acting like an encasing on top of cables, basically. That's what that would be like. That perineum, not only does it protect the fascicles, it also controls movement of molecular material in and out of these cells. So it will determine what can go inside the fascicle and help neuronal function. So that's another purpose of that. And when they zoom in on these fascicles, one big distinction you will see between the abdominal portion of the vagus nerve versus the cervical portion of the vagus nerve is that there's far fewer of these dark circles inside, meaning far fewer of those myelinated nerve cells, the nerve cells that have that protective layer that allows them to send information ultra fast. So big difference. And then, so maybe let's talk about that right now. What is the difference? So apparently the vast majority of nerve cells making up the autonomic nervous system, you're including your vagus nerve, so the system that is responsible for your reactions such as breathing, your heart rate, and how your guts function. Most of the nerves used in the control of that system are unmyelinated. So around 80% of all of these cells, all of these nerve cells that travel through the vagus nerve are unmyelinated. So remember, as I was telling you, myelinated nerve cells increase speed of how electrical impulse is sent across the cell, meaning basically, in this case, electrical impulse is like a form of sending information. And so in this case, when you have unmyelinated cells in your nervous system, that electrical impulse is slower. And so it might seem counterintuitive. Why would you want your nerve cell, so a vagus nerve cell, to act slower instead of rapidly. After all, it sends such rapid information back and forth between your body and your organ. And the reason for this is because technically, by slowing down that movement of electrical information being passed from one area to the other, it allows the vagus nerve to absorb information from other areas and nuance the ultimate final signal that reaches, say, the brain, or if the information travels from the organs to the brain, or vice versa. So it allows the vagus nerve to be much more sensitive actually to the environment and incorporate immediate changes all the time as information is really relayed from various body parts into your vagus nerve. So it's actually very clever design system, and as a consequence, the vast majority of the nerve cells are unmyelinated in the vagus nerve. Very, very cool, I thought. And we're going to talk about what is the final purpose of myelinated nerve cells in the vagus nerve as well. So stay tuned for that as well. And now when they looked at the under electron microscopy for the abdominal portion of the vagus nerve, basically you still see those dark circles around myelinated neurons as well, but you see far fewer of them and way more unmyelinated cells, way more Schwan cells. And again, they this allowed the authors now to basically compare the cervical component of the vagus nerve versus abdominal component of the vagus nerve, and how they differ in terms of the content. How many myelinated cells do you have in the top of the vagus nerve versus the bottom of the vagus nerve? And they use computational program that allow them to see this, so they were able to, literally the computer can surround the myelinated, and they used, I believe, blue and yellow to show myelin nerve cells in one area, and they showed orange to show all of the unmyelinated nerve cells, and it allows them to calculate how many you have in one area versus the other. And to give you an example, in that cervical component of the vagus nerve, when they studied that slide, 15% of nerve cells were myelinated versus, I think it was 19 that were unmyelinated in terms of area, surface area of the slide. So how much area was devoted to one type of cells versus another. But when you get to the abdominal vagus nerve, only 1% of the nerve cells in those electron microgram images were myelinated versus I think it was 22% were unmyelinated. So complete change. Most of the nerve cells are now unmyelinated. So again, remember the percentages were how much area, surface area, was devoted to these nerves in these images. So big difference right now. So that shows you that as the vagus nerve travels, it sends all these unmyelinated nerve cells throughout the body in order to have that nuance control. And this allowed the authors to actually tabulate how many nerve cells were present in their samples and how many of them were myelinated versus not. And get this, based on their calculations of the number of different donor samples that they studied, it turned out that approximately, the vagus nerve is made up of 110,000 neurons. 110,000. Now that's an average. They said there's a huge variation between different people. So one vagus nerve is not equal to another, okay? And by the time they got to the abdominal, so that was in a cervical component of the vagus nerve, 110,000 neurons to start with. By the time you get to the abdominal version of the vagus nerve, that trickled down to about 82,000 nerve cells. So, and about 80% of all of these nerve cells are unmyelinated. So that's the first paper. I'm going to actually tell you about one more super cool way of how another group of scientists looked at nerve cells of the vagus nerves, because that used fluorescent microscopy. Super cool as well. So fluorescent microscopy is when you have when you target something on your slide with an antibody is made to target something specifically. It will attach itself to it, and that antibody carries a fluorescent tag. So now you can look under specialized microscope that will detect that fluorescence, and you can see with a color. You always use green or blue, and you can combine these colors on a single slide, and that allows you to see at least 2 things per slide in terms of using a fluorescent tags. And this basically can allow you to investigate with imaging like that. And in these particular authors, in this second paper I'm quickly going to tell you about, is what they did is they use red fluorescence to target neurofilament. Remember I was telling you how nerve cells cytoskeleton is made out of microtubules and neurofilaments, so they were targeting neurofilament proteins that make up the neurofilaments, and that allowed them to basically see all nerve cells. And then with another antibody that was fluorescing green, they were targeting that myelin layer protein, one of the proteins making up the myelin layer. So that allowed them to see how many of these nerve cells, which will show up as red, are green because they have myelin around them. So very, very cool way of doing this. By the way, they also used the abdominal, they looked at cervical and abdominal portions of the vagus nerve as well. And the abdominal one was also from donors. So this movement of like looking at this in this way in terms of donors is catching on now. So that's good. But the one that's cervical was from a cadaver. But nevertheless, if you look at the image, cervical component of the vagus nerve, the one that is closer to your neck, is on the left side. On the right hand side, you have the abdominal one, and you can see on the left hand side that when the nerve exits your brain, you see tons of myelinated nerve cells, lots of green over the red nerve cells, right? And then once you get to the abdominal version, you barely see any green, right? You can see most of the nerve cells are unmyelinated, and there's very little green color. We have a big wind coming up. Hopefully that's gonna be okay. But what's very cool is that this allowed the authors, they also looked at the thickness of the myelin layer nerve cells. And here's the interesting part, because what is known is that myelinated nerve cells that have very small, thick layer of myelin, or medium thickness myelin layer, those are the nerve cells that send information around the body that influences your immunological response. So remember, we made a video describing the cholinergic anti-inflammatory pathway. It's also called the inflammatory reflex. Those are the nerve cells, the one with very little amount of myelin, that influences specifically that immunological response. And what these authors were mentioning is that in that abdominal portion of the vagus nerve, the vast majority of these very few myelinated layer are the ones that have the very thin layer of myelin. So they are there to stimulate the immunological response. And that makes sense. Obviously, your gut has to have a lot of immunological response in order to protect the gut, right? So that was really cool. Then one more thing that I also mentioned is that based on calculations of how many nerves they did, they calculated that the cervical component was only 50. So compared to the other nerve, vagus nerve that we just discussed, this one had only about 51,000 nerve cells using the technique that they use with immunofluorescence. 51,000 nerve cells at the cervical component of the vagus nerve versus 20, I think it was 3 by the time we got to the abdomen. So very different numbers, and that's the whole point. Vagus nerves, how they are made, can be dramatically different from person to person. And this finally brings me to the conclusion and to tell you why some people might not be responding to vagus nerve neuromodulation device. And the reason why is because there's all of these components that I've been telling you about can influence how resistant the vagus nerve is. So for example, how many fascicles you have, how thick is that protective layer around fascicles as well, how many myelinated cells are there, and basically all of that can increase resistance to how the electrical impulse travels, and as a consequence you might need a different energy input into your ear to target your vagus nerve, and some people might not be responsive to such information, electrical impulses, as others, and that can explain why some people might not see even any benefits potentially, because one vagus nerve is not equal to another. All right, this is one of the coolest stuff I had to study, but made it for cool, but hopefully very long, but very cool video for you. The last thing I'm gonna mention is, once again, if you're interested in vagus nerve neuromodulation device, I am sponsored by a company that released one that apparently is certified, because I haven't seen the certification. I read the information on the website. It's certified for the European market. So this is important, as you can see, because obviously when you're stimulating your vagus nerve, you're stimulating big, powerful biologic component of your body. So that's the attractive part, is that this at least is certified for, and this is new era of bioelectronic medicine. So it's very exciting times. If you're interested in this device, again, the discount code is in this video description, so check it out. Please give us a like, please subscribe to the channel, please follow the vagus nerve series. We have a playlist as well for a lot of videos on this topic. I get to study a lot of science related to vagus nerve. Absolutely love it. So check that out. Leave us a comment, and also check out our Patreon channel, where we provide additional content that does not make it to the YouTube. And I'll see you next time. Bye, everyone.